Now, the next logical step is to understand why spatial normalization is so important for the segmentation process and why segmentation and spatial normalization are usually intertwined problems. You will also learn what an atlas space is and how voxel-based morphometry has been based on these principles. Now, the general idea is that having a norm space or atlas space can help you to improve the standardization of anatomical information and help you to improve segmentation. So in order to look up the most probable segmentation result for a voxel, so-called tissue probability maps in a normative space are provided. In this example here for four tissue classes, for example, gray matter, white matter, CSF, and non-brain areas. So we know that brains share certain macroscopic general shape features, gyral patterns, and also general location features, but only to a certain degree. So there's individual variation. And to optimally use atlas information, you have to take the individual brain to the atlas space in order to look up what the most probable tissue class of that spot or voxel would be. In other words, you need a spatial normalization algorithm that takes you from the individual brain to an atlas geometry. To perform such a spatial normalization, you basically need three ingredients. So first, you have to make a definition of a 3D reference frame in which each coordinate is referring to a specific anatomical region. It's a kind of artificial brain that doesn't exist in an individual, but that is a kind of projection space. For this projection space, you need a specific template. So an image that is geometrically in exactly that shape of your atlas space. And of course, you need an image registration or normalization algorithm that transforms the individual image to the atlas space. On the right side, you can see different examples of T1 weighted templates that are in the so-called M and I space. These spaces have a long historical development, ranging from individual atlas brains to averages, for example, of 305 individuals, and different types of spatial normalization procedures behind these atlases, and so on. So if you report on your procedure, you always have to give the details of such templates. In its simplest form, you can only use rotation, sharing, translation of the image, and zooming to try to adjust your individual brain to an atlas brain. However, in most cases, you also need more local warping, so-called nonlinear transformations. And it's the combination of both approaches that eventually leads to a good match between your individual brain and the atlas space. Here in this example, you can see how adding these nonlinear steps can, for example, improve the correspondence of the ventricular space. So these yellow arrows still show quite marked differences between the fourth and the fifth individual, whereas adding nonlinear warping steps gives you a better agreement between these brains. So as segmentation and spatial normalization are kind of intertwined challenges, there have been different solutions to this. On one hand, there are algorithms that first try to segmentate the image, and then they use these first generation results to normalize these segments to the atlas space. Alternatively, you can also first try to warp the entire brain to an atlas space and then do the segmentation. There's one additional problem called the low frequency image bias caused by scanner and coil features of the MRI system. So typically the areas in the center of the brain are darker than the outer regions. And so this drop of the signal has also be taken into account. There's one very successful algorithm called unified segmentation that has managed to integrate these three challenges into one statistical framework. On one hand, this bias correction, the spatial normalization, and the tissue segmentation. This algorithm that's become quite popular for voxel-based morphometry can also analyze various MRI input types, not only T1 images, but also T2 images or EP images. So when it comes to these different coordinate systems, the situation can be quite confusing. So the so-called world coordinates would be absolute coordinates in the MRI room, for example. Whereas when it comes to the body axis as a relative reference system, you have so-called axial planes that separate inferior and superior compartments. You have coronal planes that separate anterior and posterior, and you have sagittal planes that separate left and right. The so-called stereotaxic space here of a brain is one of these mentioned 3D reference spaces, also called atlas space, that is of an artificial normative size, and that is used as a destination system for the spatial normalization steps. And as soon as you are in stereotactic space, the voxel coordinates, 3D coordinates, should actually refer to the same anatomical structures across individuals. Here you can see Jean Taillarach, who first developed this idea of a spatial standardization of, in this case, neurosurgical procedures. So he used the post-mortem brain of a 60-year-old woman to develop a certain geometric system, alignment system using the anterior and posterior commissure of the brain to put it parallel, to create a parallel plane to the ground, and then also bring it to a normative size. And from this original Taillarach space, the Montreal Institute of Neurology spaces have been developed in a stepwise way. So on the left side, you can see such a measurement grid in Taillarach space that can be used for simple morphometric operations like counting how much volume of these voxels are covered by, for example, the temporal lobe. which in the end gives you first volumetric measurements. And as explained, it was this space that is then developed to the various MNI templates. So there is a history in these templates and sometimes also a certain circularity because defined populations were used to create a template, and as soon as you reuse these templates, you kind of inherit a certain part of the information. There's a very nice web page that explains the development of these atlas spaces. Now we are very close to voxel-based morphometry, because if you manage to extract a gray matter map on the one hand and also have it ready in a normalized space, you are able to compare this information across individuals and to perform statistical comparisons. However, there's one step needed in between, because if you perfectly align the shape of the cortex or the gray matter map, you might, of course, also reduce inter-individual variability. In other words, you might make the brain so similar that you wouldn't find any differences anymore. And to account for this warping step, there is another step needed, referred to as Jacobian modulation. It's a statistical step that re-enters the volume change that you produced during the warping into the final voxel. So in the end, a voxel that is brighter in the normalized image indicates a higher original gray matter volume. And these modulated maps can then be compared statistically, mostly after adding a bit of averaging procedure over the image, a step called spatial smoothing that basically uses a defined kernel, mostly a Gaussian kernel, to average the surrounding information of a voxel. The reason this smoothing is done, it can compensate for a certain imperfection of the spatial normalization, and the smoothing also improves the statistical properties of the model. After you've done this, you can start to do group comparisons. And here on the right side, you can see an example of the before mentioned Huntington's disease. So here you can see a statistical T-map comparing a group of Huntington's patients with a group of control subjects. And as expected, you find a hotspot of the volume loss in the bilateral chordate nucleus. There's also a variant of voxel-based morphometry referred to as deformation or tensor-based morphometry. In this technique, you are not projecting your original image to an atlas space, but you rather prefer to warp it onto a later image acquisition. So it's an ideal technique to detect very subtle differences across time. In this case, you can even detect by looking at the images that there's a slight increase of the ventricular spaces over 7 months in a patient suffering from severe schizophrenia. And if you then analyze the so-called Jacobian determinant map rather than the volume map, you can directly conclude from the expansion of the ventricular space that is encoded in this deformation map that there must have been a volume increase. So in the end, it's a very sensitive indirect way to perform volumetry. And as explained, it's very useful for longitudinal studies. Now, the next chapter is dedicated to surface-based segmentation. So far, we've talked about segmentation being basically based on a decision per voxel. So each volume element of an image is assigned to a certain compartment. When it comes to surface-based segmentation, the principle is a bit different. Here, you use the original image information to create more statistical geometric objects, so-called surfaces or planes, between borders of or between tissue compartments. So one plane is representing the border zone between white matter and gray matter, and the other key plane that we need is the border zone between gray matter and CSF. As soon as you have created such three-dimensional surfaces, you can also use these to normalize your brains. And during this procedure, you flatten the surface and actually get a combined image of both gyri and sulci. Here is another more zoomed in version of the same principle. So surface planes of the pile surface and the so-called white surface that separates gray matter and white matter. And naturally, it's a clear between these planes, you can measure the cortical thickness. Here is a specific example of a free surfer segmentation. In yellow, you can see this white matter surface, and in red, you can see the pile surface. Statistically or geometrically, these surfaces are organized in little triangles. The corners being referred to as vertices of these triangles, and between these vertices, you have the edges. As mentioned, you can then also perform statistical operations and group comparisons or correlations on these flattened out surfaces. In this example, you can see blue colored areas that represent some significant results in an example comparison. Now, again, opposing these two segmentation techniques, on the left side, you can see that one important feature needed for regional morphometry is a parcellation. Parcellation means that you assign your brain regions to certain anatomical labels. The labeling system of free surfer, for example, that is referred to as the Desikan-Giuliani parcellation, is actually based on a manual delineation of a number of example brains, and it's fully automated, so it dissects your cortex into anatomical regions. Actually, the full list of gyri known in the human brain. So as soon as you have these surfaces, you can also create more complex markers. For example, the gyrification index that will I explain in a later slide a bit more detailed, or you can look at how strong the curvature of a certain region is. So if a plane surface is flat, or if it's bended in a certain measurement volume. You can also see in the left lower image that as soon as you have these two surfaces with corresponding triangles, you can also directly perform micro volumetry of such a pair of triangles. So there are several ways to come to volume information also from the surface-based segmentation. Now the next chapter is on two sets of features that are more complex three-dimensional features. The first one is based on the morphology of the sulci. The sulci are formed during the embryological and fetal development. And there are several forces driving this. On one hand, there is tension coming from axons that develop, and they pull together the basis of the gyrus. And there's also pressure coming from inside the brain, pushing out the subplate and trying to move tissue into the gyrus. And there's cortical growth, which also expands the gyrus and also it creates mechanical forces that fosters the generation of sulci and gyri. So again, the same principle displayed on the right side. In the early subplate phase, you have a low fiber content and organization, and this becomes then more and more organized with fibers developing into the gyrus and also fibers into the orthogonal direction at the basis of the gyrus that also exert mechanical forces. So these processes are highly complex and in part genetically driven, are very phase dependent. And so people found it quite informative to also analyze these features in the brain from MRI images. So what you need to make quantitative statements on sulcal measures is you need the before mentioned parcellation of the brain. So you have to assign your sulci and your gyri. So in this example, we are deep diving into the inferior frontal sulcus, which is plotted here in yellow, that connects the middle frontal gyrus and the inferior frontal gyrus. You also need an additional artificial plane, I would call it, that is called the enclosing surface. It's a plane that connects the tips of the gyri. As soon as you've defined these surfaces and as soon as you have your parcellation information, you can start to create, for example, measures of the width of the sulcus, often based on several measurements, not a single measurement. The same is true when you want to make a statement on the depth of the sulcus. You can create distances from the deepest point of the sulcus up to a number of points of the surface or enclosing plane. This is an example of a study on aging phenomena in the morphology of sulci. So you can see kind of three-dimensional transparent projections of sulci. You can see a typical matrix you could extract from these sulci. For example, the length of a sulcus, its depth, its width, as mentioned, and also its surface area, which would be the area of the surface at the enclosing plane. This is an example of a study we did in our lab a while ago. The question here was, is there a genetic difference for a specific genetic variant that encodes for worse or better computational abilities? Actually was a study on dyslexia and dyscalculia, but in this analysis we focused on the intraparietal sulcus that is plotted in red in these six individual brain examples, and using a fully automated sulcus identification software called Brain Visa, we then demonstrated that the so-called risk genotype in this case was indeed associated with a lower sulcus depth and a lower sulcus volume. We used the central sulcus, which is plotted in green, as a comparison sulcus to gain more specificity of this analysis. So you can see sulcus measurements can be quite useful for genetic questions. This principle has been taken to much higher numbers in this publication, where UK Biobank data were used to reconstruct all known sulci of the brain, and also extracting length, surface area, width, and mean depth as the main metrics. And it was demonstrated that there is a strong genetic influence on these measures, and that these measures also correlate with clinical markers to a certain degree, be it cognitive performance or disease status, for example, of Parkinson's disease, ADHD, or depression. That takes me to the next complex 3D feature called gyrification index. The gyrification index is a measure that you gain for a certain brain region. It can be any subregion you define in your tool. It can also be a whole hemisphere. It can also be the whole brain. It can also be a lobe. And within that region that you define, you calculate both the length of the pile surface, so the surface that really crawls across the gyri and into the sulci, and put it into relation to the outer contour. That is, again, this enclosing surface that I mentioned before. So the higher this ratio is, the more folded and the more complex the folding pattern is. And the lower this value is, the less folding you have. You can, as mentioned, calculate this for a certain area and then move this area across the whole brain, and by this generate a full brain map of this gyrification index. So it's quite a algorithmically complex thing to do. And as soon as you have such a whole brain or whole surface map, you can correlate it, for example, with age. And in this example publication, the authors demonstrated that particularly the frontal areas show a decrease of the gyrification index with age, which is quite interesting, knowing that the frontal parts of the brain are, of course, evolutionarily younger. This gyrification index not only declines with age, but it also seems to decline slightly different in certain psychiatric conditions, for example, bipolar disease type 1 or schizophrenia. And here I've kind of smuggled in a newer analysis technique that's also using more complex three-dimensional information, in this case, information from networks. This analysis is referred to as structural covariance analysis. It has been first used in a more prominent way in the work of Seeley et al., published 2009, where they demonstrated that certain hotspots of neurodegeneration, for example, Alzheimer's disease or semantic dementia, are showing these atrophy patterns not in a random way, but they show it in a way that resembles resting state networks, which are shown in yellow here. Now, the interesting idea was that you can detect these networks, or these network patterns, also when you analyze the covariation of gray matter volume across subjects. So rather than taking a time series, you can take a series of subjects and detect these green networks here. And this principle led to what's called source-based morphometry, which basically uses independent component analysis to extract such independent networks in a sample. We did this trying to find out how many of these independent sources, source meaning component, actually exist. So it seems to be between 100 and 200. We estimated it to about 150. And as soon as you know this source or component structure of the brain, you can also use it, for example, for clinical comparisons. You can see an example comparing schizophrenia with control subjects, dissecting the voxel-based maps into, on one hand, component loadings per subject and the actual component maps. So it's a typical ICA representation of the data. There are certain advantages to this technique. I think it's quite attractive to preserve the network character of brain changes. As we know, the brain is organized in networks, and analyzing it at a voxel level can sometimes disrupt this principle. You also have a nice feature space reduction, which is interesting for machine learning, for example. There are also certain disadvantages of looking at the data in this way. For example, you have a lower spatial specificity. So it's not so easy to then extract at which part of the network the differences actually occur. You rather get an information on the entire network. There's one important statistical issue when it comes to analyzing sizes, distances, shape features, or volumes of the brain. And this is that most geometric features scale with the size of the intracranial volume or the size of the total brain. There are several ways to define this global measure. The most neutral one is probably the volume of the intracranial space. So as soon as you have this value available, there are several ways to use it in your statistical analysis. The first approach is referred to as covariate approach. You simply add ICV and by this correct your factorial or predictor analysis by this covariate. You let it compete with the other covariates such as age and sex. The residual approach is not applied in the final model, but it is applied to your specific feature and you get a corrected feature, in this case, a corrected volume. This method subtracts or adds a certain amount depending on how strongly ICV correlates with this feature in the entire study group. So there is a certain information flow from the group to your correction. This can be considered an advantage or a disadvantage. I would rather see it as a disadvantage because the correction step is dependent on features of other individuals. A third method is called the proportion or ratio method. Here you simply divide your original volume by ICV. This can be useful because you perform the correction only within the individual. It can be problematic if there is no correlation, particularly no linear correlation with ICV, because then you are doing a kind of overcorrection. So probably approaches 1 and 2 are the most conservative ones that I would recommend. We should also take into consideration that all these approaches assume a linear correlation between your local feature and ICV, whereas these relationships also contain non-linear or allometric relationships. In the last slide, I would like to demonstrate you two innovative MR techniques. One is a specific, more recently developed sequence referred to as neuromelanin sensitive MRI. It's a T1 sequence that is very sensitive to depositions of neuromelanin, and this neuromelanin is produced as a kind of side product in the biochemical cascade that leads to noradrenaline and also dopamine. So in the examples on this slide, you can see 3 individuals with a brightly appearing locus coeruleus area at the posterior edge of the midbrain. And it comes out a bit more clearly if you average these brains and overlay them. This technique has proven quite successful to monitor aging processes, as it has been shown that the average noradrenaline turnover decreases with aging. And this decrease may be accelerated, for example, in Alzheimer's disease. But you can also quantify dopaminergic centers, for example, the VTA or the nucleus accumbens or the substantia nigra. On the right lower side, you can see a 7 Tesla midbrain slice. It's clear that with this high field strength, you have a lot of tissue contrast with more gray intensity steps differentiating the different brainstem nuclei. And I think these techniques that are increasingly available for humans will also lead to a certain renaissance of morphometry of the brainstem, for example. That is, of course, a key center for various neurodegenerative diseases. So with this, I come to the summary. Morphometry in neuroimaging refers to measuring distances, surfaces, volumes, or more complex 3D features or intensities in a common spatial anatomical reference system. Segmentation of the MR image into tissue classes represents a basic step. Spatial normalization, closely convolved with segmentation, is useful to generate corresponding anatomical frames for secondary analysis across subjects. Then, as we demonstrated, a distinction can be made between volume-based and surface-based segmentation pipelines. And both of these provide global, regional, and local measures with different biological implications and, of course, different degrees of functional predictions. What I haven't explained in detail, but it's kind of intrinsically clear, is that at all these levels, you can then perform correlations with clinical or biological, including genetic information. And of course, all these measures can be used either in univariate or in multivariate analysis. Thanks a lot for your attention.